Great. All right. Well, good afternoon, everyone, and welcome to Grand Rounds in Team Science, or GRITS. I'm Sandra Lee, and I'm Professor of Medical Humanities and Ethics and Chief of the Division of Ethics here at Columbia. I also have the privilege of leading the TEAMS resource as part of the CTSA at the Irving Institute of Clinical and Translational Research. In the CTSA, we recognize that addressing the complex challenges of clinical and translational research requires intentional efforts to cultivate and sustain high-functioning interdisciplinary teams. TEAMS, which stands for Team Expertise and Management Support, is a resource that fosters collaborative, inclusive, and high-impact team science by providing guidance in a range of team science domains, and those include team composition, communication, synergy, conflict management, and sustainability. And as part of the team's work, we've launched the Team Science Award of Distinction for Clinical and Translational Science, which recognizes teams conducting groundbreaking research who have engaged in successful teamwork and that develop innovative approaches to collaboration, mentorship, and knowledge sharing. So today we have the privilege of learning from our awardees, our inaugural awardees, that constitute a group of exceptional researchers who have demonstrated outstanding commitment to interdisciplinary collaboration. This year's honorees, the Maternal-Child Research Operations Team, or MACRO, have made extraordinary contributions to maternal and child health research through their highly interdisciplinary work, led by investigators from obstetrics and gynecology, pediatrics, and psychiatry. MACRO has successfully integrated clinical and translational research. Their collaborative infrastructure includes the OB Commons database and the Perinatal Biobank, and has provided an essential foundation for advancing research on pregnancy outcomes, fetal development, and early life health interventions. Among their notable initiatives is the COVID-19 mother-baby outcomes, or COMBO study, which has provided crucial insights into the effects of SARS-CoV-2 on maternal, fetal, and infant health. This research has had a direct impact on clinical practices and public health strategies, and illustrates the power of collaboration across disciplines and departments. So as we move into today's discussion, we will hear directly from our panelists about the strategies, challenges, and innovations that have shaped their collaborative research. Their work really does serve as an exemplar of how team science can drive forward meaningful discoveries and improve health outcomes. So following this, I'm going to introduce our 6 panelists. Really want to thank you all for joining us today, and I look forward to a rich and engaging discussion. You can always add questions into the chat. I'll be monitoring them as we go through all of the presentations so that we'll be able to hopefully get to as many as possible, and then we'll have about 30 minutes, I hope, for rich discussion after the presentations. So now it's my great pleasure, and I'm going to introduce all 6 so that we can seamlessly go through the presentations. But I'm going to introduce all 6 of our awardees. And the first is Dr. Ronald Wapner, who is the Director of Reproductive Genetics and Professor of Obstetrics and Gynecology at Columbia. Dr. Wapner is an internationally known physician and researcher specializing in reproductive genetics. He has authored or co-authored over 450 publications and has been an active investigator in the area of maternal-fetal medicine. Dr. Wapner is the co-founder of the MACRO. Our next speaker is Dr. Anna Penn, who is Chief of Neonatology and is a physician scientist. Dr. Penn's lab studies the role of placental function in fetal brain damage and cross-disciplinary research that she has dubbed neuroplacentology. Her work provides new opportunities to understand the role of placental hormones in shaping fetal brain development in ways that may lead to neurobehavioral disorders, including autism spectrum disorder or ASD, and suggests that early treatments based on placental hormonal replacement might ameliorate the risk for ASD and other neurodevelopmental disorders. Dr. Penn is a co-founder of the Macro. Our third speaker is Dr. Catherine Monk, who is the inaugural Diana Vagelos Professor of Women's Mental Health in the Department of Obstetrics and Gynecology and Professor of Medical Psychology in the Department of Psychiatry. Dr. Monk is also the founding director of the Women's Mental Health at OBGYN, an embedded initiative where she and other mental health professionals help women with stress, depression, and anxiety across the lifespan. Our fourth speaker is Dr. Dani Dumitriu, who is trained as a general pediatrician, neuroscientist, and pediatric environmental health scientist, and is an associate professor of pediatrics. She dedicates 80% of her time in basic science research into the neurobiological basis of resilience as a PI of the DOOR lab, or Developmental Origins of Resilience lab at NYSPI, and 20% of her time caring for newborns in the Well Baby Nursery at the Morgan Stanley Children's Hospital here. She is the lead of the combo. Our fifth speaker is Dr. Morgan Firestein, who received her PhD in psychology from Columbia and is currently an associate research scientist in the Department of Psychiatry. She is a developmental psychobiologist with formal academic training in developmental psychology and technical training in molecular biology. Her research is to identify early emerging biomarkers of neurodevelopmental risk with a focus on ASD to facilitate early identification and early intervention services for at-risk children. Dr. Firestein is the director of the Perinatal Biobank. And then finally, we have Dr. William Fifer, who is a laboratory director of the Fetal Infant Perinatal Research Laboratory and the chief of the Division of Developmental Neuroscience at NYSPI, and the associate director of the Institute of Developmental Sciences at Columbia. He has active collaborations within the departments of psychiatry, obstetrics, pediatrics, behavioral medicine, and public health, as well as the Columbia Population Research Center. And he will be the final speaker for today. And with that, I'm going to hand it over to Dr. Wapner.
Okay, thank you very much. I'd just like to take a minute initially to thank everybody for basically giving us this award, in particular the CTSA, which is an exemplar of how multiple disciplines can come together and work. So if I can say one thing, we thought that we would start by ending, and this has been our goal. This has been our vision. We together want to change the culture by building and creating an ecosystem for cutting-edge multidisciplinary maternal-child research. If I could have the next slide, please. Thank you for the introductions. It saves me some time, but I do want to point out on this slide that this truly is multi-departmental. We have individuals from OBGYN, we have individuals from peds neonatology, we have individuals for basic science, psychology, etc. And as far as macro is concerned, all of these circles are irrelevant because we really work together as a team, and the overlapping and thought processes for what we want to accomplish is really transdisciplinary. I also want to mention that there are many other members of macro, not just us, but what you see here today and those of us presenting are basically, for lack of a better term, the steering committee. The next slide. is going to talk a little bit about history. As with most, well, this is the index. This is the outline of what we're going to cover. I'm sorry, this was added. I'm going to talk about before macro. There will be a discussion of the developmental origins of macro that'll be presented by Anna. If there's another one, we're talking about the extending the multidisciplinary research capabilities and how macro has had a significant role in doing that. The next, we're going to talk about maternal mental health and the development of origins of health and disease, which Catherine will cover. We are really proud to have Dani present Combo, which really talks about how she has taken the team science and extended it well beyond Columbia, how this started to understand the pandemic effects of the mother-infant dyad, and you will hear us talk about that dyad multiple times in our discussions. And finally, Bill is going to, I'm sorry, Morgan will give the overview of the perinatal biobank, which has been incredibly important to ourselves and has served as a resource for the institution. So the next slide. Okay. This talks about what started this. And as most things, it started based on a clinical problem. And as you see illustrated here, the problem was that we had two groups of people taking care essentially of the same patients, but they were totally separated administratively, intellectually, and in many other levels. On the one side, we had the OB department and maternal fetal medicine taking care of the mother. And in particular, although the mother's critically important, a lot of the work was based on also taking care of the fetus and the to the time of delivery. Then something magical happens. A stork drives in and moves the patient that the OB people were caring for to a nursery. Many, if not most, go to a healthy baby nursery, but also some will go to the intensive care nursery. And I used the stork intentionally to point out the problem that we had. The problem was that there was no communication between the two. Yes, there was somewhat clinical communication if we had a complex case, but there was little to no sharing of data, and they really were treated as two separate entities. At the time, that has been the way that OB and pediatrics and neonatology had practiced for an incredible length of time. But from the standpoint of healthcare, and most importantly, we're interested in from the standpoint of research, that made no sense whatsoever, because as an obstetrician, our outcomes, and we all like research that deals with outcomes, was going to the nursery and we knew nothing about it. So we weren't held accountable as much as we should be. So the next slide illustrates really the chaos that we worked under. There were many, many reasons for the chaos. One reason was the moms were considered patients in an adult hospital. The kids were considered patients in a pediatric or children's hospital. So they were cared for and they were considered not to have communication. I think the prime example of this was 10 years ago, there was no linkage between the mother and the child. If we wanted to do research in which we looked at the outcomes, we would actually have to go to two separate medical records. We would have to look at the mother's chart and then look on another sheet of paper to find out the neonatal's medical record number. And obviously, things were not ideal for the kind of research that we needed to do. The next slide is what started us. And one of the problems was data sharing. We had two separate medical records, and there was no consistent center for information. That meant for lab tests, that meant for care, that meant for a lot of different things. And one of the problems, and you will hear how we solve this and how it really has become critical. We knew that in order to do this, we had to have a new model for a database who considered the dyad as a single entity that could be studied. Now, this could be done in a number of different ways. This could be done as one single database where all the information goes, which many people use, or this could be done by building a federated network, because each of us have multiple different inputs. Some are outpatient, some are inpatient, some are ultrasound, et cetera, how we could find better ways to bring the information together. I also want to point out that this, even if one thinks about it, expands further, because both the OB service and the neonatal intensive care service don't just take care of people that come to Columbia initially. We have maternal transfers. The nursery is, I'd say, overwhelmed, filled with babies that deliver elsewhere who are equally important. So we knew that we really needed to have a ability to combine the data, and at the same time, the National Institute of Health started to have networks, and one of the requirements to be in the network was to have an active maternal-child database that could then be used to share information on the patients that we are taking care of. And that has continued to grow and hopefully will continue to. Next slide is what our ultimate goal was. And the ultimate goal is clear. I could not have said it more times. And treat the dyad of the mother and infant as a single entity, which for 9 months it is, and don't make arbitrary changes just because the child delivers. We wanted to do this in research. It's critical nowadays to do it in care and improving the outcomes. And at the right, probably was what, at least to many of us, was most important. We wanted to use this as a training for our young investigators so that we can train the leaders in our field going forward, and we could change and develop a model for the future. So you listen to, this is Anna and I whining 10 years ago about what we needed for all of these reasons. And this is when macro became an idea, and the ability to do all of these things began to develop. So I will turn this over to the co-developer, Anna, who will, and I just want to point out the pun of developmental origins of macro on the title.
Thank you, Ron, and thank you to the CTSA and the committee for recognizing the team science approach that we have taken. It's been one of the highlights of my coming to Columbia. Next slide. So Ron may have been whining for 10 years, but I've only been whining for 5 years, because I only arrived here 5 years ago in January 2020. But one of the reasons I came to the Division of Neonatology at Columbia was because there were already working relationships that existed that had begun to develop across multiple areas. And because I studied the placenta, that is an organ that really takes two. And there has to be a dyad to have a placenta. And so that was one of the draws in coming to Columbia. So one of the first relationships and standing meetings I set up was with Ron, and we wanted to focus initially on the training aspect of increasing maternal fetal medicine and neonatal fellow research interaction. And we came up with the idea of perinatal pizza meetings. And then everything changed. So this was not supposed to be part of my transition to Columbia, not in anybody's playbook, but by March 2020, we were dealing with the COVID pandemic. Next slide. And so then things started to happen really fast. I went back through my emails, actually, and these are actually the dates of different emails that went out. When the first obstetrical case was identified at Columbia, then we circulated the first labor and delivery and neonatal management plan, which we then revised weekly after March 13th. And then the first delivery started, where we knew that moms who were COVID positive needed to deliver, and we had to take care of both mom and the delivering baby and the staff who were then being exposed. And then on March 22nd, the obstetrical team had the foresight to start screening all women admitted to labor and delivery for COVID. And so this led to, it led to a very early publication, the first publication out of the obstetrics department, that showed that asymptomatic COVID was actually a very high percentage of the population that was carrying COVID and spreading COVID, although they weren't sick. And so this really defined, moving forward, how we managed COVID clinically. But it also, next slide, it also led to a huge scramble for COVID-related samples. The CTSA and CUB are well aware. But we needed something special. We needed the maternal neonatal dyad samples. And so we founded something with the somewhat clunky name of OB-GYN and Pediatric COVID Perinatal Research Collaborative. And the goal was to review all the proposed studies that were being developed in April of 2020 and going forward to use this population and biosamples with the intent of maximizing research potential and not overwhelming our families. Thankfully, May 6th, Dani gave us a name, which was much better, which was macro. And the executive council got established with myself, Ron Wapner, Dr. Gianfini Bannerman, Catherine Monk, Melissa Stockwell, Dani Dumitriu, Bill Fifer, and then we had support through the OB department. There were three main subcommittees that formed with many, many people involved. First was biospecimens. How are we going to collect maternal and neonatal biospecimens, store them safely, but keep them connected, which was not something that any other part of the hospital needs to do. Second, how are we going to deal with the data? And we learned a lot about the nuances of the data systems, although OB Commons was already set up. There are multiple versions of REDCap. There are different places the data exists. We had just moved to Epic, and so there were a lot of problems to be solved there. And then we had a follow-up team of really finding out what happened to these women and babies that were going to have been exposed to COVID, and we had no idea what the potential long-term outcomes would be. And that subcommittee really evolved into Combo, which Dani will tell you more about. So where are we now? We started as an oversight committee to sort of adjudicate which samples could be used for which activities, how not to approach families multiple times within a short period of time, and how to, as rapidly really build information for the world of what this pandemic was doing, how things were behaving, was there placental transmission, things of that sort. It has evolved into an organization. It's evolved into a much bigger ecosystem. And it now is something that spans multiple departments. We still have monthly steering committee meetings where we talk about current and planned research. We foster collaboration for everyone from fellows all the way through to senior faculty. We provided advice and guidance. We review protocols. We also provide resources once we've discussed protocols, writing letters of support for grants, providing biobank specimens or access to data. We're really trying to focus on trainees and young investigators, but we have seen people at all levels, and a really broad ecosystem has emerged that is focused on this dyad in terms of education, research, infrastructure, and resources for all of our investigators. And so now we're going to hear about several examples that have come out of and have been, that are part of the macro ecosystem. Excellent. So this is the team today. And this just shows you all of the different preconception, pregnancy, birth, and infancy related areas that we work in. We're working across OB, psychiatry, pediatrics. Uma Reddy has joined us. It's been a great pleasure. She is the head of our maternal-fetal medicine unit. And she came about 2 years after I did. And then the rest of the faces you will recognize as we present. And then we're going to hear the first example from Dr. Bill Fifer.
Great, thank you, Anna. So you can see we came going a little out of order, but they're saving the best for last. So let me, in the next 5 minutes, allow you either to take a little cat nap or talk to you about how we've been extending multidisciplinary research capabilities through the wonders of macro. So prior to when macro showed up on the scene, our members had many successful perinatal research partnerships. But we had many challenges, particularly with regard, as Ron alluded to, integrating pregnancy and infant clinical data and being able to put that all in one very accessible place. It was a real trial. And we still also remain somewhat siloed from other investigators with whom we could have collaborated and shared related approaches, and in particular, lessons learned, the kind of thing you don't find in publications or even in talks many times, but that's what we wanted to be able to do and do it as quickly as possible. So you're going to be hearing later about some large-scale, very successful efforts from our team, but I'm going to share just 4 slides to talk to you about current studies that are extending our macro umbrella and our multidisciplinary research capabilities. So next slide. The first example is a partnership with Pediatrics and in neonatology and psychiatry with developmental neuroscience. And if I could have the next slide. Oh, thank you. Next, just hit next, yeah. And it really is talking about our attempts to do sleep assessment and interventions for NICU infants in parents pre and post-discharge. Next. And as many of you know, we've had either family members or friends. It's very clear how poor sleep health really is the signature of preterm infants and parent life during this most critical period of neurobehavioral development. Next. Our next example is a collaboration with obstetrics and psychiatry on the development of an overnight prenatal, maternal, and infant overnight sleep in the natural environment. Next. Again, that's a very understudied phenomenon, which just only very recently can now be studied non-invasively using very novel technologies. Next. And then the last example, we won with a teamwork with obstetrics, pediatrics, psychiatry, and psychology, which is an emerging partnership with our relatively new NIH Autism Center of Excellence, looking at a prospective study of early markers in infants at genetic risk for autism and related disorders. Next. And this, as you might imagine, is just the kind of complex study that really needs an entity like Macro. Next. So very quickly, here's one example, one slide to show you some novel technologies which now allow for in-depth sleep assignment at home, which go far beyond the usual metrics of the duration of sleep and questionnaire data and measures just based on activity. So here, the actual sleep cycle now can be quantified, and with respect to the sleep stage phenotypes, how it is that these are fragmented and then emerge in some coherent patterns. And now, with wearable devices, as the ones portrayed below, which are now been validated and have now been used in hundreds of participants of mothers and infants in the natural environment, we're moving quickly forward with this endeavor. Next. Our second example displays how sleep cycles can actually be studied before that baby shows up outside the womb, and can be studied before birth with using bands that the mother wears, can wear throughout pregnancy, that measures her heart rate and activity, as well as the fetuses, and allows us to get to what we've aspired to do for so long, which is to understand how the heart and the brain work together to develop the circuitry that's going to be needed for the autonomic nervous system to do its job as soon as the baby is born. Next. And then here's just one example, naturally the best one that I could find, but showing fetal sleep cycles overnight in the home, where you actually can see in blue an indication based on movement and heart rate variability, a fetus in quiet sleep, then merging up into green, which is active or REM sleep, back again to quiet, back up to REM, and back and forth throughout the whole night for over about 7 hours. Something that, again, could never be done before, and now has been done with our team, and we're working with other teams around the world to be able to exploit this phenotype. I mean, these technologies. And next and last is an example that is just emerging now. It's our effort to perform a prospective study, as I mentioned earlier, of early markers of infants at genetic risk for autism. And again, this study requires this expertise and collaboration across several disciplines that now can flourish under this macro team science approach and culture. And next will be Catherine.
Hi, great. Thank you. Thank you to the CTSA for having us here and for the honor to be talking with you all today. So I'm addressing maternal mental health and then this concept of developmental origins of health and disease. And so really adding some more background about this dyadic approach you keep hearing about that really is a central component of all of the work that we're doing. Next slide, please. So it's probably not really news to anybody in this group here today that mental health conditions are overall, those assessed and identified, more common in women, particularly for the most common mental health conditions of anxiety and depression. Women have almost twice the rates, and that's across the life course. Next slide. But maybe what might be some new news to some of you is that depression and anxiety, what we lump together is under the term perinatal mood and anxiety disorders, again, depression and anxiety being the most common of these PMADs, they are actually the #1 complicating condition of pregnancy and childbirth. And so nationally, PMADs affect up to 1 in 7 pregnant and postpartum women. Next, please. And what you can see here, just really briefly wanting to talk about some of these disparities and outcomes, is that those who are living in poverty experience particularly high rates of postpartum depression, as you can see here, and rates of depression in general are more than double in the Black population during the perinatal period. Next, please. Oh, thank you. Yes. So Here's a quote: "We're not going to forget about the baby while we take care of the mother." This is from really a founder of the field of perinatal psychiatry. This begins to introduce this dyadic perspective. So not only in raising a child in their, their context, their social, emotional, economic environment, does that influence the child and their future neurobehavioral development, but this influence from the parent can start with the pregnant individual. And I'm going to be introducing that concept now. So next slide. So this is, again, maybe some of you are familiar with this, but a research area called Developmental Origins of Health and Disease, or DOHAD. It's also known as prenatal programming. Another felicitous phrase here is how the first 9 months shape the rest of your life. So again, you can really hear this dyadic perspective that is not only the child in their context starting postpartum, but all the way back in the prenatal period. So we can call this a peripartum dyadic perspective, pregnancy and postpartum. Next slide, please. So again, just to explain this research model rather briefly, the idea is, and adding an evolutionary perspective, is that maternal experiences function as exposures that forecast the postnatal environment to the fetus. And the fetus responds biologically with adaptations, with the aim, if you will, to improve their fitness for a later stage in development in the postnatal environment. And the risk for future health may come in part when there's a poor match between the maternal experiences and that cueing of the fetus about the postnatal environment, but the postnatal environment doesn't match the prenatal. Next slide. So very briefly showing some work from my lab. And you can in some ways just look at this as visuals, and I will walk you through it. On the left, you're seeing 3 squiggly lines, which are representing a latent profile analysis of healthy pregnant women having healthy pregnancies. Fetus is doing fine. But when we did a close characterization of stress indicators, and you can see some of the variables on the top there: blood pressure, diet, their mental health. The pregnant individuals parsed into 3 different groups. And now you can look on the right and start to see the parsing: a physically stressed group, a psychologically stressed group, and a healthy group. So the first graph there on the left, on the right, the left one, the bar graph, looking at preterm birth rates, they are far higher and exceed our national average in those with physical indicators of stress, higher blood pressure. Psychologically stressed, also somewhat higher preterm birth rates, and the healthy under our national average. On the right there, you're seeing differences as fetuses in the 3rd trimester as the heart rate and movement are correlated. This is a very rough index of some kind of central nervous system development, and you can see a decrement in that in the physically stressed fetuses of physically stressed pregnant individuals. So wanting to send home the idea that it really kind of a proof of concept just in this one slide that there is a dyad before birth in terms of maternal experiences potentially having an influence on that future child that matters for their neurobehavioral development. We know preterm birth, for example, is a risk factor, not only for neonatal mortality, but also for conditions such as ADHD. So next slide. What are the mechanisms? Many labs, as well as ours, and Anna, of course, with her looking so extensively at the placenta, have been looking at the mechanisms. One that comes up often is immune activation related to maternal stress. Next slide, please. So you can see how the pandemic would really lend itself to this DOHAD research perspective. Is it the virus that is potentially going to influence the future child if the mother is infected? What about her immune activation? Is that going to affect future brain development and brain development in utero? Maternal stress through some biological mechanism haven't yet identified well. There's other pathways that the child could be affected. So you're going to hear more from Dani on a lot of some of the outcomes from really macros first, sort of focusing on the pandemic. That's fine. I want to take a few more brief minutes to say, if we go back to PMADS and we want to keep the dyad in perspective, the rate limiting factor, the biggest impediment to recovery from PMADS is access to affordable mental health treatment. That's going to affect the mother, and it's potentially going to affect the future child. Next. So something that you've maybe seen these reports, ProPublica, it's so impossible to find mental health care in the US that's covered by insurance. Next. So something else that happened coincident with the pandemic, but what I think was very beneficial to our patients is Dr. Mary Dalton, the chair of obstetrics and gynecology, had the idea to start up an integrated mental health service, which I led and am continuing to lead in the department. And I want to show you briefly just some access to care results. Next slide. You can see that these results are from, did the date get lost? Oh, good, it's at the bottom, really from the pandemic period. And we're looking at 2,000 patients, the majority of whom are pregnant or postpartum. And we are taking insurance. So just very quickly to focus on the graph on the right there, the bar chart, you can see we have a very high utilization rate. People are coming to see us after referral and they're getting access to treatment much higher than standard rates. So affecting the mother in a positive way with downstream effects on the child, we hypothesize. And that's something more for future research. Next and last slide. So where do we want to go? Well, you know, Ron really addressed the idea of this culture change, changing the ecosystem. The mother's electronic health record, as of last week, now her storyboard is in the baby's chart 0 to 2. That's really important for clinical care and lets us think more and more about research with dyadic perspectives. So, for example, we want to also continue to change the research culture, have easy access to the electronic health record data. So you're going to hear this word a lot, universal. We can do population-based studies with this dyadic perspective with our shared OB, pediatric, and psychiatry patients. So for example, can we look at a depression trajectory? And if we have mental health intervention, we can test the DOHAD model. We should see better outcomes in those kids when their mother's depression is treated during pregnancy. I'll stop there.
And I'll echo everybody's thank you for having us. Thank you for the award and thank you for having us for this presentation. And I will take you through a little bit our combo initiative, which, as Anna mentioned, was one of the 3 working groups of our macro initiative that we began in early 2020. Next slide, please. First and foremost, it really, I want to point out that this really exemplified the team science approach from the very beginning. As an overview of the history, Catherine Monk had actually started a prenatal cohort to understand the effects of the pandemic on the maternal mental health, while I and my colleagues in the newborn nursery had started a cohort of newborns to understand the effects on the newborn. Much of the work actually across the 2 groups was led by Morgan Firestein, whom you're going to hear from soon. And in the summer of 2020, we got together, integrated into the current combo cohort or COVID-19 mother-baby outcomes initiative. Along the way, in the last 5 years, we've had numerous other members join us from both within Columbia as well as across, giving this cohort a really rich development. Next. To show you a little bit of the scope of where we've gotten to over the last 5 years, we had really rapid mobilization such that our first dyad was enrolled on May 26th. To date, of 2020. To date, we've enrolled about 2,000 mother-baby dyads, and we actually were able to expand into 2 other states, Utah and Alabama, through a CDC contract that Dr. Melissa Stockwell, another member of the macro committee, had secured in order to study surveillance of SARS-CoV-2 infections in pregnant women. We were able to therefore also enroll some of those pregnant women postpartum with their children in order to study their outcomes. We have about 80 unique instruments, survey instruments with about 27,000 unique variables. And to date, one of the most innovative pieces of combo has been our video or telehealth visits, where we, for example, do full neurodevelopmental assessments via Zoom. We've had about 24 publications to date, many in high-impact journals, secured over $15 million in funding. And most importantly, we offer a wealth of training opportunities. We've had 3 T32 trainees, 3 K99 trainees, 2 of whom were awarded, one including Dr. Morgan Firestein, who will be presenting soon, and many, many undergraduates and master's students have completed their theses with us. Next. To give you an example of one of our early outputs, our very early focus was on vertical transmission, as it meant so much for the world at the time and in the spring of 2020. And this was a beautiful collaboration between OB and Pediatrics. There was over 40 of us that got together, and very rapidly, and I mean very rapidly, within 14 days actually from conception to submission to the journal, we put together the data on the first 101 babies born within our hospital system, looking at the rates of vertical transmission and confirming the safety of moms and babies rooming in postpartum, as well as safety of breastfeeding. This was cited by the CDC and influenced the turn of national and global hospital policies away from mother-baby separation, which was happening at the time very early in the pandemic, as we didn't have enough information. But as we've heard, the dyadic approach and in particular, their early relational health, which is the focus of my own research, is really important. And so this helped global policies move away from separation. It also led to a Dear Colleague letter that Senator Gillibrand put forth in August 2020, which ultimately was one of the pieces that helped infuse the $1.5 billion Recover effort. Next. Another early output has been to look at the neurodevelopment of babies born at the height of the pandemic. So as Catherine mentioned, the DOHAD concept was big on our minds in both pediatrics and obstetrics and psychiatry early in the pandemic. And in particular, we know that other viral infections during pregnancy and other immune activating experiences can lead to adverse neurodevelopmental outcomes. So we put together data on babies born in 2020 and showed that in fact, COVID infection in moms during pregnancy did not have an effect on the child at 6 months, but just being in utero during the pandemic and presumably through the stress that the mother was experiencing, there were slight risks to neurodevelopment. Small in terms of not frank neurodevelopmental impairments, but nevertheless possibly important at a public health level. We also, just last fall in September 2024, had a paper come out on looking at autism risk, which has been on the minds of many of us. And just to give you the punchline there without showing you the paper, we did not find an increased risk of autism with either exposure to maternal SARS-CoV-2 infection or just being in utero. We are continuing to follow this cohort. Right now, for example, we are looking at risk of early childhood psychopathology, which we do not yet have a full story of, but we do have some early indicators that possibly there is an increased risk as a function of COVID infection. Next. Most importantly, this has led to the design of a cohort that is truly scalable and nimble. We, in the very beginning, just focused on getting early data, right, postpartum, And then since then, we've been chasing this cohort as these children have gone older. In the fall of 2024, we just launched our 4-year assessment, which is ongoing. And we've also been able to add components to the study such that we've grown into a really deeply phenotyped cohort. We added electronic health records as part of an NIH supplement. Of course, our bread and butter has always been surveys and video visits, but we've also done really innovative things like ecological momentary assessments and, in a subset, EEG and EKG, olfaction testing in mothers, as well as we are NIH funded to do brain MRI scans of both moms and babies. And this has allowed us to now actually move into another phase of combo, where in the next few months we are about to start a national expansion with many of the universities that you see on the bottom, towards a pilot, initially in a pilot phase, but towards a cohort really focused on early relational health as the main outcome. Next. You're not supposed to be able to read this, but this is just one of our infographics of the numerous teams that have benefited from data in combo. We share freely. We are always happy when groups come to us and are interested in joining. Next. But just to give you a small flavor, here is one of the interesting outputs that has happened that really I would have never imagined when I started combo with 2 groups from ENT. One overseen by Jonathan Overdevest, looking at maternal olfaction, and this was a paper that was recently accepted, and one looking at infant hearing, led by Anil Lalwani. Both of whom came to us for completely different interests than the initial cohort, but both of them very successful collaborations that have led to actual meaningful research outputs. Next. And now I will hand it over to Dr. Firestein to tell us about the other successful work group.
Great. Well, thank you once again to the CTSA. Thank you so much for this opportunity to give a brief overview of our macro perinatal biobank, or the PNBB. This initiative was really first started by Dr. Wapner and Dr. Penn several years ago, and it was established to support the study of maternal, fetal, and placental factors, as we've been discussing today. And I've been really fortunate to be involved with macro, initially as a contributor to combo, as Dr. Dumitriu just discussed. But I also, like Dr. Penn, am particularly focused on the placenta. And so over the past several years, I had firsthand experience understanding the need for this kind of repository. And so I've really enjoyed serving as the director of our perinatal biobank since 2022. I want to just quickly share some of the primary goals that we considered when establishing our biobank. So the first is to collect and process biospecimens collected during the perinatal period to support a wide range of research applications. And the second being, of course, to responsibly store and distribute those biospecimens to investigators. One of the things that we've really spent a lot of time trying to address is how can we ease burden on patients? So this has been brought up earlier today, but trying to, as much as possible, minimize the potential for a patient to be approached multiple times by multiple independent investigators. And similarly, how can we reduce redundancy in the collection of these biospecimens, and even thinking to future assaying of the samples? So to give you a brief overview of the timeline, we first launched our phase 1 in August of 2022. And this was a period where we were very focused on engaging the clinical staff. This is predominantly clinical staff in the OB/GYN department. And it was during this time when we enrolled our first participant. Then just about 1 year later, we launched phase 2, where we shifted to a model where we were largely engaging the OB research staff. So these are full-time, very well-trained clinical coordinators who are able to screen participants for the biobank, obtain consent, and collect and process samples. And I'm very pleased to share that just last month in January, we met our phase 2 recruitment goal of recruiting 500 pregnancies. And I'll talk a little bit more about the specific samples that we collect, but to give you a sense, this has resulted in the collection of just about 14,000 samples since launching phase 1. So we do our best to enroll our participants prenatally. This can be in any trimester. And upon recruitment, we collect a sample of maternal blood, maternal urine. And we are then able to utilize some of the existing infrastructure through EPIC to actually track the progress of these pregnancies, and our staff are notified as soon as that patient is admitted to labor and delivery so that we can subsequently obtain linked samples at the delivery time point. So again, we get a repeat sample of maternal blood and maternal urine. And we also are able to then collect cord blood and placental tissue. So I just want to emphasize, consistent with everything that has already been discussed today, that one of the really unique features of our perinatal biobank is that it inherently involves the mother, the child, and the placenta. And so it really takes this truly triadic approach, necessitating attention to all three. And the second unique feature is that we have been very intentional in the design of the perinatal biobank, such that we're able to very easily now link maternal and child charts and samples, which historically has been quite difficult to do because there just was no mechanism in place to do this with the prior infrastructure. And just briefly to give you a sense, our samples are collected for, as I said, a range of applications, including analyses of RNA, DNA, proteomics, hormones, inflammatory markers, lipidomics, and many, many more. So I just wanted to also show the distribution of the samples that that we have, or the specific conditions that are represented in our biorepository. So you can see that the majority of the participants are classified as having had an uncomplicated pregnancy, but we do also very specifically recruit for conditions including gestational diabetes, gestational hypertension, preterm birth, fetal growth restriction, known diagnosed or suspected fetal anomalies, recurrent pregnancy loss, maternal or fetal familial history of a neuropsychiatric condition. And finally, we also try to co-enroll participants who are already enrolled in other studies that are collecting overlapping biospecimens, so that additional samples can be collected into our biobank. Oops, sorry about that. The one other point I want to make here is that if you add up these total numbers, it exceeds the number of patients that we've actually enrolled into our biobank. And that is because many of our participants fall into more than one category. For example, preterm birth and fetal growth restriction are likely to have significant overlap. But this is actually a great strength of our biobank because it affords investigators the opportunity to study comorbid conditions as well as individual ones. And then more recently, we have also initiated a longer-term follow-up component to our biobank. So given that so many of our macro team members study infant and child neurodevelopment, we felt that obtaining this kind of longitudinal information about child development was really crucial. And so a third unique feature of our perinatal biobank has become this implementation of a long-term parent report infant follow-up assessment. This is done at 1 year of age. It is automatically sent to participants when the child turns 1. And what's really wonderful is that investigators are now able to directly link these longer-term outcomes to data from health records and, of course, to our samples. And so looking ahead to phase 3, our next goal is to try to think about how we can move our model to one of universal collection of birth specimens. And there are many reasons that we're interested in doing this. Of course, we are interested in increasing the inventory of our current repository, trying to make it more representative of our larger patient population. But even more broadly, we really hope that this will complement Macro's future plans for universal tracking of mother-infant dyads. And Dr. Dumitriu will speak about that more in just a moment. I was just going to show one way that we have tried to prepare for this model of universal collection is in developing what we call these rapid use research kits. So these are very carefully curated research kits that match our current collection protocols very directly, but they can be easily implemented by clinical staff. So in an event where our biobank staff are unavailable to collect the sample, we can also engage the clinical teams to assist with that. So I do want to turn it back to Dr. Dumitriu, who will give a brief summary of some of the next steps that will really further our overarching mission to support the multidisciplinary research at a larger scale.
Thank you. So as you've heard over the last half an hour or so, we have made really incredible advances in Macro on follow-up and on biobank. But there was a third, if you remember early during Anna's presentation, there was a third team that we had early on, which was data science. And this has been probably the most challenging piece, as virtually everybody on this presentation has pointed out. There's really unique features to dyadic data that are not accounted for by in other data sets. So I am very pleased that Dr. Paul Curtin has joined us as an associate professor of computational data science and pediatrics just as of January 1st. Within the center that we established in the fall of 2024, the Center for Early Relational Health, which the official announcement hasn't come out, but will come out sometime in the next few months. Next, please. Dr. Curtin is really well-versed in leading the data science, the data core for our center, initially really focused on integrating all the combo data that we've acquired. Again, we have so many sources in terms of REDCap and EPIC data, video data, MRIs, many, many modalities. So his initial focus over the next 1 to 2 years is really going to be to integrate the combo data set with the vision of taking that into a much larger data set that really combines maternal and child data. He is very well experienced in this arena. He has led multiple NIH-funded data cores, including for CHEER, which united data sets from over almost 50,000 individuals across 50 R01s. Next. As well as similar work within industry, where he led data cores for example, FDA projects. Next. And in his short time with us, Dr. Curtin has really started to educate us about the concept of big data versus smart data, which is where we want to move towards. So in the past a few decades, we've really focused, and in particular in combo, we've really focused on this concept of bigger is better. But bigger is not always better because it does not maximize the data velocity of getting from data to ideas or results. And so he's really focusing on building an infrastructure where you can very easily and intuitively integrate across these different modalities and get results very quickly. Next. And in particular, the focus of the data core that we are building is very much aligned with the NIH strategic vision, which I'm sure many of you here are familiar with in terms of data management and sharing and following FAIR guiding principles. And we don't have data on this. This is really at the very beginning. This work is really just getting started, but we did want to just mention this as a plug for a place where we're very interested in joining the larger community, especially of the CTSA, to really think closely together on how to collaborate on this topic. And I will turn it back to Anna to close us out.
So thank you everybody for participating in this. Just to take it home, we've had a lot of successes. Combo has been a true highlight. We've developed a strong structure for collaborative support. And I think a number of people on this call have presented to Macro, and I hope we've been useful to them. We've developed the perinatal biobank. but we still have many opportunities to go. Next. We, as Dani said, are just at the beginning stages of setting up universal dyadic database, as well as a universal dyadic biobanking system. Next. But this is only going to be possible through partnerships. And we really want to discuss with the CTSA and with others here, and of course, across the city and across the country, how best to leverage the community that we've developed into long-standing partnerships that can help with both funding, research, and training. So I thank you for your attention today, and I think we're open for discussion.
Thank you. That was tremendous, really amazing work. So I congratulate all of you. If we could take the slides down and maybe see each other, we do have time for questions. And I am going to start us off, but folks that do have questions, feel free to put them in the chat or to use the raised hand signal, and we can get to your question quickly. So I wanted to ask you a question about some of the learnings at the institutional level. I've been really just so impressed in hearing all of your presentations on your ecosystem approach and how you went about it in terms of solving specific problems. But I am struck by, I think it was Dr. Wapner who started it off by saying that there's certain infrastructure institutionally that just is not conducive to interdisciplinary research, despite your successes. And I see that you all are probably now experts in our own institutions at CUMC. What are some of the things that you have learned about challenges and opportunities at our own institution for this type of interdisciplinary work? And anyone can jump in.
Yeah, I think the challenges are not unique to Columbia. A few of them are, but most of them are not unique to Columbia. And that has to do primarily with access to various data systems and to some of the broader efforts that are going on that simply aren't built to link mother and baby and long-term follow-up data. I mean, it's been true at the NIH level for the last 30 years. There is the maternal fetal medicine system, and then there is the neonatal research network, right? And until a few years ago, there was no effort to try and connect the two. Then there became some effort to connect the two, because MFMU studies stop at the point of delivery and NRN studies start at the point of delivery. Well, that makes no sense, right? We need to connect these things. And so I think a lot of it is just the divide that is natural in most fields, right? You have one MRN number and you can track that person for many, many years, and you can track their samples. But having the two go together is far more complex. I also sit on the CUB committee that approves sample uses. So I get to see it from that side too. And the concern is always, are we going to deplete the samples, right? Which is very legitimate concern. But for us, it goes beyond that. We want our samples used for paired research. So we don't want to be in the position of having someone use all of the baby samples and not have gotten the maternal samples to match, right? And so that becomes that becomes much more of an ecosystem understanding of the problems. Data sharing is the same, and there are definitely hurdles between Columbia and NYP that are unique in terms of medical data sharing and access to data. And I think Dani can address some of those. I saw Nim Goldstrom on earlier, who is one of our neonatologists who has been working with Ron and Dani to develop ways of pulling data in a more rapid manner. I think those are the types of things that need to be tackled and will benefit anybody who does translational or clinical research. But we are a peculiar subset, but we'd like to share our experience born out of necessity, and form some real partnerships now.
Wonderful. Any of the other panelists want to add anything to that?
I would love to just jump in to point out that there's something really unique at Columbia in terms of the crosstalk between pediatrics and obstetrics that I think is quite different than most places in the country. In part, that is, as Ron mentioned earlier, our physical location, being that we take care of moms and babies in the same place, but it is much more than that. We routinely ask each other to be on each other's papers to ensure accuracy. So, just down to the nitty-gritty, when we categorize maternal complications to look for baby outcomes much later, we talk to our OB colleagues in order to make sure that the categorization is done properly, and vice versa, which I think is truly unique in this field. There's often, papers within our work that are led entirely by OB teams or entirely by pediatric or neonatal teams. And we are very unique in that we always seek each other's input.
I would just add also that consistent with, a dyad in a sense being about a relationship, I think a lot of this kind of success, which, is, Dani and Ron and Anna have been significant, I have to say, leaders, and Morgan, amazing. I've been so honored to be part of this team, and Bill as well. But the relationships that go back quite a ways, that then build more and more networking. And so I think Columbia is huge, and I know, and I agree with you, other institutions have very similar problems. We're not unique. Like Dostoevsky, all families have, their crazy problems in different ways. I think Ron brought that up in one of our meetings. But I've always felt, and it's been nearly 30 years being at Columbia, like a kid in a candy store. So on the one hand, just amazing opportunities for connecting with researchers across disciplines and people, almost uniformly being incredibly interested in collaborating and sharing ideas. But I think forming those relationships and then keeping the momentum going and having the wherewithal and the persistence for some of the impediments, which are just there structurally, and they're not, they're not purposeful, but they just make things complicated, and continuing to chip away at them and chip away and chip away. And then I think you see something like macro really growing into the kind of ecosystem culture change that we're really starting to see in research and clinically.
Great. One of the things that grew out of those relationships that I've been so impressed with since coming here is, we have built macro financially on a shoestring. And everybody's pitched in a little bit, right? I didn't have much to give. I gave a little. I didn't have, much extra floating around obstetrics. He gave what he could, and then negotiated for, some of the clinical research coordinator time. Dani is building up this database program now. All of those things move us forward, and the sum is more than the total of parts. And so I think we just need to keep doing that on a bigger scale now.
Great.
Murdoch.
Yes.
Yeah, I'd just like to, first of all, congratulate the team. I think this is, probably a first of kind under Sandra's leadership to celebrate and acknowledge teams in this way and to evaluate them and have a competitive kind of process for this, which I think is always really interesting because it makes it brings a little bit of self-awareness and reflection to individuals and teams, like why are we so good, or why are we achieving this or not achieving this together. So as part of that, the first thing I want to do is just congratulate you, because this is a super team, and when you looked at the teams, when we looked at the teams that were part of the process, this one had, you know, many unique and wonderful characteristics, and you've really presented them well today. As you get to this point, and as you sit here today, and you know, as you've gone through the process, putting the application in, reflecting on why you're a team that does well, and highlighting that, I'm hoping has also led to the opportunities to think about how to go to the next level. And, you know, you showed some of those examples, and you were doing that anyway, but just, you know, humor us here, you know, maybe the award and Sandra's initiative has helped with that. And I'd like to ask two questions, or two related questions. Like, one is, you know, this is an interesting time right now, obviously, with change of administration. Yet I feel that there's probably tremendous opportunities also in the space that you're working in, because it's a challenging one. It's one that's, you know, you're uniquely positioned to address questions, and there's a lot of health challenges there that are not well addressed. So the first question is really, what are you thinking about is the most important things to be focused on as you go through the next year, for example, because it is a challenging time. And second, I see Reddy sitting here, and Uma co-chaired a committee work group with Richard Mayhew on how to bring together at the school and medical center level, you know, moving to the medical center level now, more investigators, particularly junior mid-career investigators, to have access to the investigators and the cohort studies that are unique and special at Columbia. I think Melissa was on that committee as well, so that you can build more capacity through expanding the pool of researchers that can actually use the data. And you pointed out some like amazing ENT faculty who just are amazing, actually, the way they kind of are able to move in these different directions. But you know, I'm just really interested in how you see and how you'd like to draw people in to increase the ecosystem. And maybe that's something you talked to Uma about, you know, because of our role on that committee as well. But we're very interested at the school level of how to bring people together, like really bringing people from different disciplines, because you've now made your data kind of searchable, the summary level data searchable, and questions and answers can be directed to investigators. And then there's support to try and get people to do pilots and then to go towards bigger grants. But what do you see as the next year, and how would you see you're kind of building the ecosystem and bringing people in while not having people cherry pick, you know, the samples that using the samples in an appropriate manner?
We actually, since the beginning, realized that was an important part of what we wanted to do. And both within our departments and outside of our departments, we developed a system when people wanted to do research in this domain or wanted systems in this domain. They learned about macro and they came to us and we asked them to present a concept. And then all of us commented on the concept and gave them permissions. Because frequently, if somebody from some other discipline comes in and wants to do work in this area, they're not familiar with a lot of things. So we have found that by bringing them to our group, by our group serving as a resource, they've come in with some ideas, left with entirely different ideas of how to do it. We have given them other individuals in our field who could work with them. So we actually have served as that kind of resource, and it's worked really well. We have had blood samples go to different people. We have had a number of different things. So I think the personal touch is really important. And in answering the questions, and I'd be interested if others agree with me. I think one of the things that has made us really successful is we started at the grassroots. We had a problem, and that's exactly how we continue to function. Somebody comes in and says, I need this to be successful. Another person comes in, it doesn't have to be in the executive, and then we problem solve. And that's very different than a top-down approach in which somebody says, this is a solution for everybody. And frequently, then when there has been a solution for everybody. It hasn't matched goals with what we needed to accomplish, which had a little bit of a role of why we felt that we needed to do this. So I really think the combination and the communication between the grassroots investigators, understanding their needs, allowing them to give comments and input on how the bigger systems are going to be developed is really important. And I can't tell you, and this is not, I can't tell you, and I think the data system has been a prime example. There have been multiple things that one wanted to do using existing operations in which the answers were, you can't do this, you can't do that, that's not consistent, that's not consistent. And Dani was forced and successfully to say, okay, how am I going to do this for what I really care about? And then I think what we said is 100% true, that we would love to be part of the development of the bigger systems, but this really has to be flexible and responsive to each other's needs. It just can't be, this is what we're going to do, make your system work. Now, that's my opinion. I could be a different voice. I don't know what others think about that.
Well, I would love to pitch in a little bit more in terms of what we're envisioning in terms for the data core. Murdoch, the examples that I gave are beautiful, but between the investigators coming to us in combo and saying, do you have this data, and us being able to actually provide the data, it was at least about 9 months. In another case where it was the actual NIH funded work, it took us over 3 years, way after the NIH funding ended, to actually integrate the data set necessary for what we had proposed. So this is a critical step in terms of next steps for the next few years to integrate that data in a way that isn't, just like you said, aggregate data is easily available, but also, and I shouldn't speak for Paul, he's writing an R1 for tomorrow, so that's why he can't join, but the way that he's envisioning this data core is that it will also be you'll be able to actually get data very easily by simple language. So how many women gave birth between March and August of 2020 who had children that were premature, and then the system would be able to generate that kind of aggregate data very easily. And therefore, the access to the data will be enhanced many fold. I think that's clearly the next step. Of course, for us in combo, but also as Ron and Anna have alluded to, it would be great to have that at a bigger level once we get the proof of concept within combo.
One of the things that, just to add to Dani, you know, NIH comes out with NOSIs and then they come out with RFAs. And as part of writing the grants, the turnaround time is incredibly short. And you need your pilot data, you need that information, and there needs to be a way to get that. And I think some people use data navigators, but that goes through a system that may take significantly longer than we have to do it. It's not writing papers. Of course, we all want to get things out as quickly as possible, but there is a continuous urgent need to do that. There are some very good institutions where investigators at their desk can at least get data within a very short period of time, at least metadata to write the grants and accomplish what they want to do. So each of us have lost grants because we couldn't get data in the timely fashion that we need. So that's just another reason. Now macro can do that. I could call, now I can, not macro with combo, I could just call Dani and, she says, you're a pain in the neck, which she says to me all the time anyway, but I can get you that data by tomorrow. It's a very different, flexible, responsive system that we really needed and is what we're building.
I think just to build on both Ron and Dani's comments, it really is about the relationships, because what we need to do in the next year or two is develop some really strong infrastructure grants. And those are probably the most difficult grants to get funded, because everybody says, yeah, we'd be happy to give you money to do the research after you build the infrastructure. And so we've cobbled it together. And one of the reasons I think we've gotten this far is, you know, Ron and I felt very strongly about the biobanking. So we took a little bit of money and supported Morgan. And Dani feels very strongly about solving her database problem. She is invested in doing that, but she's open to having everybody else pitch in with ideas, with effort. He has basically adopted one of my neonatology faculty to really bring in the perspective of the OB Commons and the follow-up data sets as well. And so, you know, once the system is built, it should be able to be flexible enough that it can solve multiple problems. So I think, and that just has to do with relationship and the fact that we meet every month and that we all talk to each other and that we share.
And I
Bill,
just
did
quickly
you-
wanted to add to that, if I could, Senator, that what you've done today, I think, is let a number of other people know that what we're doing and what we're interested in doing, that maybe would like to cooperate with, or rather to work with us in ways that it extends just not macro, but just this whole culture and approach. And is if we can do that and get all these clever minds, I'm sure, that are around here on the screen right now, to be able to think, oh, wait a minute, I think I could do that. And I'd love to talk to a group of people about it who are trying to solve these kinds of problems. So I wanted to sort of end with the idea, we thank you for at least getting the word out and having people be able to think about, geez, maybe I could do this if I just sit down with some like-minded people.
Okay, well, this does bring us to the end of our time together. I just want to really express gratitude to all of you for your tremendous work and for really being an exemplar of what we're trying to achieve through team science. You justly deserved this award, so congratulations. And thank you also for your presentations and for the audience for engaging the work. As Sheila mentioned, this recording will be available on our SharePoint, and a link will be sent out via email. So if you have colleagues who would be interested in hearing about macro, I hope you will send that along to them. And I just wanted to let you know that our second Grand Rounds in Team Science is occurring next Tuesday, March 4th. So in about a month. 12 to 1:30. It's a collaboration with the VPNS Office of Research. It will be on authorship. The title, Who is First Author? Negotiating Authorship and Research Teams. I'll be giving that talk, so I hope I'll see many of you at that event. But I wish you all a wonderful week, and thank you again to Macro for being our Team of the Year.
Thank
Good
you.
seeing everyone.
You.